In this video, we will discuss about how the dendritic cells help in dissemination of SARS-CoV-2. In the previous video, we have just touched upon this idea that dendritic cells helps the SARS-CoV-2 to infect at the intestinal tract. So, in this video, we will discuss how they have made that point. So in order to and the idea comes from the function of the dendritic cells, that it in the case of HIV virus, like dendritic cells helps the HIV virus to decimate at different parts of the body. And uh, from that from that observations, the idea comes into the researchers that dendritic cells would also helps the SARS-CoV-2 virus to decimate across the intestinal tract. And so they starts by comparing that this whether this SARS-CoV-2 virus or SARS-CoV-2 pseudovirus can attach to this or bind to the dendritic cells or not. And along with the dendritic cells, they also did the experiment on the Langerhans cells, which are found on the skin. And these dendritic cells are in vitro differentiated from the monocytes. So in this case, we can see that the dendritic cells can bind to this SARS-CoV-2 pseudovirus. However, this binding will be inhibited in the presence of this low molecular weight heparin, or in the presence of this heparin, or low molecular weight exosarpine. So in that case, the binding get reduced, and same things they have observed for Langerhans cells, and which are the cells that are derived from the skin. And when they check when they check the infectivity of this SARS-CoV-2 virus on the dendritic cells and the Langerhans cells, they can see that in the case of dendritic cells and Langerhans cells, the SARS-CoV-2 presence are very low, or pseudo SARS-CoV-2 virus presence is very low compared to the positive control case. And same things they have observed for and the reason till now they have observed that SARS-CoV-2 virus can bind to this dendritic cells and Langerhans cells. However, they do not infect these dendritic cells or Langerhans cells because they lack ACE2, which is essential for internalization of the SARS-CoV-2 virus on the dendritic cells. And after that, what they did is they check whether this ACE2 they check whether these ACE2 expressions are present on a dendritic cells and Langerhans cells, or and the next cells that they have used is this HER7 to 7.5. And in that case, in all the three comparisons, they can see that in the case of HER7.5, the ACE2 expressions is significantly higher, and in the case of dendritic cells and Langerhans cells, they do not detect any kind of ACE2 expression. So thereafter, they design an experiment. In this, they need to check whether dendritic cells can helps in decimating the SARS-CoV-2 virus at a site where the cells has the ACE2 receptors. So this is what they have designed the experiment. That is, they incubate the dendritic cells or these Langerhans cells with SARS-CoV-2 virus. Then they wash and remove the unbounded virus, and then they co-culture those dendritic cells and Langerhans cells along with this HER7.5. And this HER7.5 has this high expression of ACE2 that is validated by this panel D. And what are their hypotheses? Their hypothesis is that SARS-CoV-2 virus will decimate from the dendritic cells to this ACE2 expression cells, epithelial cells, or this HER7.5 cells. So this is their idea. So till now they have validated till now they have validated the point till this point 2, that is SARS-CoV-2 virus can bind to these dendritic cells. And after that, they co-culture these dendritic cells. After washing, they co-culture these dendritic cells along with this HER7.5 cells. And in this case, they can see that SARS-CoV-2 infection on these HER7.5 cells when they co-culture with dendritic cells is significantly higher compared to the case of this heparin and axonine, because in the case of heparin and axonine, the binding of the SARS-CoV-2 virus decreases. That results in the low infection rate. And the same thing they did it for these Langerhans cells also, and they find the similar kind of results. That is heparin and axonins. In the presence of heparin or axonins, the infection reduced down because the SARS-CoV-2 binding to these dendritic cells is low compared to the case where there is no heparin or axonin. The SARS-CoV-2 infection is very high from this Langerhans, from the co-culture of these Langerhans cells to the Her7.5 cells. So from this they made uh, they made a point to strongly support the hypothesis, that is co-culturing of these dendritic cells and Her7.5 cells, uh, or the dendritic cells that are already bind with the SARS-CoV-2 virus. When they co-culture with this Her7.5 cells or the ACE2 expressing cells, it leads to the transfer or decimation of the SARS-CoV-2 virus from dendritic cells to this ACE2 expression. And finally they validated this hypothesis on the actual SARS-CoV-2 isolate, and in that case also they validated the SARS-CoV-2 infection through this PCR, and they can see that in the case of these dendritic cells with Her7.5 they can see the high SARS-CoV-2 expression compared to the case where exoparin is present. And similarly they see these Langerhans cells with this Her7.5, and in that case also they can see the SARS-CoV-2 infection is very high compared to the case of this hexosarapine, as hexosarapine is the inhibitor that prevent the binding of the SARS-CoV-2 virus to the dendritic cells or Langerhans cells. So from this they made a point to support their hypothesis that dendritic cells can help in decimation of SARS-CoV-2 virus to a place where there is high ACE2 expression receptors containing cells are present. So I hope you got an idea about how this dendritic cells helps in the decimation of SARS-CoV-2 virus. If you need to understand more about this experiment and results, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.